Hello everyone, my name is João Meneses. I am a junior research fellow at the Nanosafety Group. I am going to present an interactive and dynamic scoping review which explores the relevance of extracellular vesicles in nanotoxicology. The goal is to understand if nanomaterials modulate cellular responses mediated by extracellular vesicles. Let's focus on the concept of cell communication. Recently, extracellular vesicles were identified as novel cellular communication mediators. Such fact is supported by its cargo, which includes DNA, RNA, and proteins. Such extracellular vesicles are released by the donor cell and may induce changes in the physiology of the receptor cells. Due to the exposure of humans to nanomaterial via inhalation and ingestion, their accumulation and difficult elimination have been raising concerns about possible harmful effects. Let's imagine a real-life scenario where a child is eating M&Ms. As you may know, the bright colors of M&Ms are due to the presence of titanium dioxide nanoparticles. The cells of the gastrointestinal tract will be in contact with those nanomaterials, raising possibly adverse effects. So we can divide the research question in two parts. Are nanomaterials able to cause abnormalities in the extracellular vesicles cargo by the donor cell? And if the extracellular vesicles cargo changes by nanomaterial stimulation, is there any abnormal response on the receptor cells? In this work, we systematically map the data available in the literature. Databases such as PubMed and Web of Science were consulted, using the following terms: extracellular vesicles, nanomaterials, cellular responses, and modulation of EVs. After removing the duplicates, 2,944 articles were obtained. The next step was to identify the most relevant studies. The first stage involved screening titles and abstracts. The second stage was to screen full-text articles based on their relevance. A total of 18 studies were included in the final review. To complement the previous workflow, two co-occurrence networks of the most frequently used keywords in the selected studies were obtained. Regarding the networks, each circle represents a keyword and each color represents a cluster suggesting a similar topic among the publications. Overall, both networks enhance that nanomaterials exposure affects extracellular vesicles biogenesis, microRNA cargo, and protein expression. All of these terms are widely involved in cell communication. The main biological activities referred to in the 18 articles were angiogenesis, biogenesis, and cell communication. After understanding the selection of the 18 most relevant studies, it is time to summarize the biological outcomes of each article. The classical way is to present such information by a table. As a complement, we obtained a dynamic and interactive network. To do so, we took advantage of the table structure, namely its headers, elements, and common connections. The obtained network is the following one. Firstly, the network may seem too complex. However, the best way to analyze it is to search for strategic points. For instance, we can focus on the cellular outcome, such as secretion of extracellular vesicles that is related to its biogenesis. Here, we can see that 15 out of the 18 selected studies, nanomaterials were able to stimulate extracellular vesicles biogenesis. Another example is nanomaterials elimination, where we can check the presence of gold nanoparticles, ions, and single-walled carbon nanotubes. Thank you for your attention.